So I'm just going to also allow the individuals online, but if anybody wants to start with a question.
Question for you, Ryan. Sure. We were in this room almost 365 days ago. You set out obviously similar kinds of objectives that we have seen here today. Yep. What's your feeling on the last 365 days of progress?
I think the capital markets have been extremely tough for us and for other companies. As a result, the execution of certain objectives gets delayed. I think the other delay that the company faced was when we met with the FDA in July 2023. They had, and it's their right, to say that they wanted to see some more information. So they specifically wanted two things, two primary things. One is what happens to the patients after 450 days? So do they fall off a log, or does the duration of the response continue? The second thing they asked is central pathology review from our local pathology results. So they seem like easy asks, but to do that, you have to rev the clinical protocol. You have to get that approved by Health Canada and the FDA. You have to then send it to all the clinical study sites. They have to put it through their research ethics board, which has to approve it. Once they approve it, then you have to reconsent all the patients. And some of the patients are available, some are not, because it's been a few years since you treated them. So they spend a lot of time hunting down and chasing patients. So what happened is, because they're doing all this work to be in compliance with the FDA, which is ultimately the goal, right? You don't want to treat all your patients and then come down to approval time and they say, well, what about these two things? And now you're going to get turned back on your heels. So the company elected to go and be in compliance with the FDA. As a result, that slowed down the enrollment of the patients. But I think that's changed in first quarter 2024. I think we're back in around 52 patients, probably at this time last year. We're up to about 68. And the data is all quite strong, and the clinical study sites are moving forward. Now, that's not to say all of them. Some of them are still behind on enrollment. Some of them are doing really well. We have some clinical study sites that are in double digits as far as patient enrollment. So I think that the company's done extremely well. Based on Matt's presentation, you can see that we sit extremely well as far as our clinical data set with the majors, and we're at a fraction of their market capitalization. So I think there's certainly an opportunity as the company goes forward and people start to see the news and hear the news for us to get value in the organization.
Quick follow-up. The request for central pathology, is that an unusual request by the FDA?
The FDA is a weird duck, okay? They say in their guidance it's recommended, or talk to our group about whether it's required for your study. What they really mean is what they should say, this is mandatory, right? So they're the FDA and Health Canada regulators, when you're dealing with them and talking with them, what they say is only what's in the guidance, how it's written exactly, okay? If you ask them something outside of the guidance, they'll say, we don't have an answer for that. It will be a design review question. Submit your data. We'll look at it at that point, and we'll give you a decision. Now, those decisions cannot be favorable, because they may say, well, we've decided that you need this. So the recommendations are always mandates from the FDA. The FDA put out their guidance in February 2018. It's now June 2024. So it's probably 6 years old. So through the two approvals that they've done or three, they've done Keytruda, they've done Adstiladrin, which is the Ferring drug, and they just recently did Immunity Bio, which is the Antiva drug. So from those groups and meeting with those groups, they've come out with what they'd like to see. So they don't put it in writing, and they don't tell anybody. It's only when you go talk to them. So I think it was a bit of a surprise for us that they insisted on central pathology. because the concordance level, which is how local pathology compares to central pathology, is extremely high, 95 to 100%. I think when I was talking to Dr. Peter Black, who runs the UBC site, he did a study for a monoclonal antibody. I think it was Atezolizumab was the drug that he did, Atezolizumab. And his concordance on his 120 patients was 100% across it. Why? Because local pathology, you have board certified pathologists reviewing the data. They're experts in their field. Okay? You're now going to take the slides that they're looking at and give it to another board certified pathologist who also is an expert in their field. The amount of time that these two men or women would disagree with each other, virtually zero. Because they would say, we don't have enough data, or this is cancer, or it's not, because they're making decisions on patients' lives. Whether you're cancer free and that's not a problem, or we think you've got cancer and you should have your bladder removed. Because there's a 12% morbidity, and I think almost a 14% mortality risk from the surgery, from radical cystectomy. So these decisions aren't made easily. So I think it was a bit of a weird ask because the concordance is extremely high. You can imagine if you go to your doctor and he says, yes, you have cancer, and you go to another guy, he says, no, you're perfect. That's probably not going to happen. Right? So it was a bit of a weird ask, but there are the rule makers, so they're the judge, jury, and executioner for where we want to go. Because the ultimate goal is FDA approval, because you want access to the US market. They're the gatekeepers, so you're going to have to comply with what they say. Do you have another question, or is that good?
What role does Canadian regulatory play in the process? Obviously, the FDA is very important in the US approvals, but.
Well, the Canadian market is a tenth of the US market, so obviously it's not as big. The US market is the biggest in the world by far. And I think that they must control 25 to 50% of the world market, depending on what specialty you're in. Canada follows the FDA guidelines almost to the letter. And so if you ask Health Canada a question, they say, well, please refer to the FDA guidelines, right? So that's what they follow. And I think a lot of Western medicine has advanced so much that I think the world economies follow it. China follows it, India follows it, Russia follows it, Europe follows it, Canada follows it, everybody follows it. So if the US FDA is asking for this, it's something you want to comply with because it will cover you off for a lot of the other countries, which is the main objective. Bill.
I'm just going to take, sorry, Bill, I'm just going to take a question here. Just a quick note online. Questions will be given priority that are not anonymous. They're good questions still, but we're going to give priority to the questions of people who actually put their names behind them. That's great. Thanks. So let me just take a look at the first question here. This has not been answered here. Just a follow up.
Okay, Christopher Horner. I know you can't speak specifically, but with all of TLT's amazing science, has there been any interest at any form or at all from potential pharma partners for foreign or domestic? Many companies at early stages have such partners. If TLT has no such prospects, why do you think that's the case? I'm sorry, Chris, we can't answer that question. Next question. Given the difficulties and stats with BCG, is there a chance Rutherin could become the new standard of care for NMIBC? I think that with ruthenium combined with BCG, which is a lot of the work that Dr. Mendel's lab has done, the data has shown that it improves the response of the molecule based on zeta charge. Okay, cancer is negatively charged, BCG is negatively charged, so they're going to repel each other. Adding ruthenium, or Rutherin, I should say, to BCG switches the zeta charge, which is the charge on the outside of the molecule, positive, therefore making it attract like a magnet. I think it has huge potential, but again, that has to go through clinical studies. You'd have to take that, you'd have to show if you're doing an intravesical installation, It's now a combined new molecule. BCG is approved, Rutherin, hopefully we get that through in GLP talks. We'd have to speak to the regulators about whether we can combine it and instill it, or what studies we'd have to do that work. But you'd also have to do clinical work. So this would involve time and money, right? Which is the company is focused on commercialization of its primary asset. I think they would be better as a partnership. Is that good for you?
If we can go back to the
Bill, sorry.
Yeah, go ahead, Bill.
There was talk about IV, and I'm just wondering, is there somewhere or another you can identify maybe a cancer that hasn't been found through the IV?
I don't know if I'm qualified to answer that question.
Maybe some doctor does.
I think that there's two doctors in the room. I think that to say that there's a disease or a cancer which hasn't been identified, I think cancer can affect a lot of different cells. And I think on one of Matt's slides,
Right.
he talked about 200 different types of cells,
Right.
and over 100 types of identified cancer. I think that the work that Arkady and I plan to do on GLP toxicology later this summer or the fall will provide what's called a human equivalent dose level for injection into the body. If a patient has a specific cancer, then that drug will localize to the transferrin receptor sites of that. If it localizes to a space which hasn't been identified, then that may be able to be picked up on some sort of radiological thing. Do you have a response, Arkady?
Yes, I do. Just to follow up on that is, would it be advantageous to put a marker on this so it goes run your blood? For a marker?
Yes and no. Markers are good in the sense that if you put like a fluorescent marker or a magnetic marker or things along these lines.
You don't need a marker. The drug harms tumors. So technically, if the drug harms to tumors, we can assume that if the drug will be injected intravenously and doctor has some suspicious lesions in the body, the biopsy would be taken from this lesion, benign or malignant, we don't know. But we know that the drug will get only to malignant and not stay over 24 hours in normal tissue. So therefore, if the tissue is taken, in theory, one can run specific mass spectroscopical analysis and to find Rutherin inside the tissue. If Rutherin will be identified in the tissue, that means it's highly likely the patient has cancer. If it doesn't, it's highly likely patient doesn't have cancer. It's whatever tissue was taken,
Including the bone marrow?
including the bone marrow, because the drug should get inside bone marrow.
That's the 24 hour period.
Yes, within 24 hours period, you can identify the drug inside the tumor cells, and actually in some tumor cells it falls over.
Yeah, so in follow-up to Arkady's response, radiological way would be the way to go, because if you start putting markers onto molecules, you start changing their characteristics. And that would involve more research, more pharmacokinetics, pharmacodynamics. So probably not the best idea. Another question?
Sure. Grab one here from Michael.
Okay, Michael Bashiro. Why do some patients respond well to this treatment while others don't benefit from this treatment? That's a fair question. I think that why do some people respond to certain drugs and other people do not? They have physical characteristics which are different. their disease, like bladder cancer and cancers, come in many different types, stages. Um, they've had different treatments. They have different concomitant diseases. They have, if they've gone through BCG or if they've gone through TURB procedures, they may have scar tissue. They may have damage to the bladder wall, to the urothelial wall. If you have disease underneath scar tissue, it would be very hard for the drug to interact with it. We have seen where there's upper stage and lower stage disease. So the disease can hide in the ureter, can hide in the urethra. On men, it can hide in the prostatic urethra. We've seen diverculi on bladders where the disease is inside the diverculi, meaning another small opening on the side of your bladder, which would affect it. So I think that what's proven in the technology, if the drug gets to the cancer and is light activated, it will destroy it. That is what we've proven. If the drug doesn't get there because it's either deeper or in a location it can't get to, or if the light doesn't activate it, that'll be a problem. We have seen where there's disease in the bladder neck, where the bladder neck is very close to the urethra. The bladder neck may be shielded by the cystoscope, the metal cystoscope. So the drug may get there, but if it's not light activated, it won't work. So as long as the drug gets to the disease and is light activated, it will destroy it. Is there another question?
Yep. We can do this one here. First they didn't name.
Anonymous attendee. I'm incredibly excited about the prospect of Ruvidar having a positive impact on non-small cell lung cancer. But I'm concerned with how we will be funding this initiative. How will you be funding this study while funding the funding for the bladder cancer study? Fair question. The company has gone out on press release and stated that we're doing a non-brokered private placement. Well, actually, we haven't done it. We've done it under a Form 4A. We're doing a non-brokered private placement up to $10 million. The company had a burn rate last year of approximately $4.5 million. As a comparison, Immunity Bio, which just got their approval a few months ago, has a burn rate close to $500 million a year. So we're a 1% burn rate with a significantly better profile and clinical data. With the company plans to do what's called a base shelf prospectus. In order to receive a base shelf prospectus, the company must demonstrate that it has 12 months of cash flow in the tip. So you can't raise the money and then say you have 12 months. In order to be base shelf eligible, you must have the 12 months. So if the company had a burn rate of 4.5 million in 2023, based on the financial statements you were given, as long as the company raises over $5 million, we should be in a good position to be base shelf eligible. Once we're base shelf eligible, we can apply to the Ontario Securities Commission for a $100 million base shelf. Once we receive receipt, what it's called, from it, and they're going to ask some questions, but once we receive the receipt on that, the company would have access to $100 million over the next 25 months. So it's not that we have the money, but we can raise the money fairly quickly based on milestones of the company. So the idea is to do the small raise, the non-brokered private placement, which we're doing now at 22 cents with a 30-cent full warrant for 5 years. If we're over the $5 million, we can become base shelf eligible. We'd put up a $100 million base. With that, we would use that to draw down funds to fund not only to complete the bladder cancer study, but also for brain cancer, for lung cancer, and for the liquid cancers that we've talked about, blood-based cancers, leukemia, lymphoma, and multiple myeloma.
Perfect. I guess there's a good follow-up for that as well, since it's a lot of financials.
Okay. Anonymous attendee. Can you comment on how you plan to acquire funds to continue this trial and how and other preclinical and clinical studies? What are the non-dilutive options? So I've just talked about how the company plans to raise funds. The non-dilutive options, there's two specifically. The company can use debt, okay? One of the issues with using debt is asset matching. Because if you use debt, let's say we borrowed $5 million or we borrowed $10 million and we spent it, we'd have to pay that money back. Plus, we'd have to pay interest on that money. So it's not that we're going to take this money and buy an asset, such as an apartment building, or we're going to buy gold, or we're going to buy stocks and we're going to hold on to it. This is research dollars we plan to spend. So from an asset matching point of view, debt is not really the best choice. The other option is a royalty deal for a non-dilutive option. In a royalty deal, you would talk to some of the royalty companies that are out there. The biggest is in New York, called Royalty Pharma, where they buy your future revenues or a slice of your future revenues. So Royalty Pharma did a deal with Ferring, which has the drug Adstiladrin, where they gave them $500 million US for 8% royalty on sales of Adstiladrin. Now, if Adstiladrin doesn't sell, then Ferring got a pretty good deal. If the Adstiladrin is a blockbuster drug, it may not be a good deal. So you're really selling off your future revenue for it. It's attractive, but usually royalty farmers only deal with companies that have FDA approval, not pending FDA approval.
Okay, perfect. I think one more on the financials, and that'll probably cover it. It's just the this one here from Anonymous right here, and then I think we've probably covered all the anonymous.
Anonymous attendee. Are there any talks or possibilities of government grants for the newest news releases? At this point, no, but there's certainly programs that are available. Usually government programs are matching programs where you put up a certain amount of money and the government matches those. The days of the governments, either the FDA or the US or the Canadian governments cutting you big checks for commercial products or pending commercial products are pretty much long gone. There are different organizations that will pay for research. There are organizations in Canada and the US that will do that. One of the risks that you take by doing a deal with organizations that want to pay for research is they, in their agreements, usually put in march-in rights, meaning that if you don't develop it within a certain time period, they have rights to come in and take it over, take it from you and develop it on their own, right? Without paying you for it. So you've got to be a little bit careful on that. But there are different government matching programs. I know of some Canadian government programs which will match 50% up to about $10 to $15 million. Yep. So there are some opportunities to work with them, but the company has to put up 50% of the money. So if you want to get $15 million from the government, you got to put up 7.5. Okay, so you're still going to have to raise money at some point.
Perfect. Any questions from the audience here? Or because we have some questions here too, so it's up to you if you want to just raise your hand if you need to add something. All right, there are some good questions, so I don't know if this is something we can really answer here. And
anonymous attendee.
Yeah.
Another one. Well, they're all the same. When will the 2-year data be released? Sorry, what 2-year data?
And then this one down here from Warren.
Okay. Warren Moriarty. Has Theralase investigated a non-Merck source for BCG for the combo treatment? No. The reason is Merck produces the Tice variety of BCG, 50 milligrams. It's the only one FDA approved. So recently, Immunity Bio got approved with their drug Anktiva to combine it with BCG, which they did to work with the Tice BCG strain. Merck didn't want to give the drug to Immunity Bio, I believe, and there is a shortage of BCG in the US. So Immunity Bio did a deal with an Indian organization called the Serum Institute, I believe, in order to get their source of BCG. The problem that Immunity Bio is going to run into, and they're probably working hard at this, is that the Indian strain, which I think comes from the Russian variety of BCG, is not FDA approved. So they're going to have to go get it FDA approved, which may be a 2 or 3 year program in order to use their drug, N803, Anktiva, for the US population. So it's a bit of a conundrum for them. Yes, they have FDA approval. I believe that they're, based on their financials, they're burning about $500 million a year, Canadian, about 380 US. They have no commercial products. So, but they have a fairly good market cap. So I think that they're going to have to make some decisions as to what they're going to do. Either they're going to have to market this in other countries, or they're going to have to work very, very closely with the FDA to get it approved, which I think is a 2 or 3 year program.
Perfect. I think we've discussed the patent lifetime that it goes to 2038. So I think that's from Anthony. Thanks for that question, Anthony. 2038, we can extend it to. Christopher again. There's a good one actually down here. This one here, but for me to first. This has been asked a couple times by this gentleman.
Scott Jarvis, can you speak to why the most recent CR data decreased across all assessment visits by some 5%? I don't know if it decreased 5%. I think what the company has done is they've scrubbed the data in order to look at the IR responses, so the indeterminate responses. When you dig down into the data, some of those have become complete responses. Some of them have become no responses. So as we build our data set, and we're at 68 patients, I think that you're going to see some patients swing the numbers down simply because the company takes a very conservative view on the data. So if a patient failed at 90 days and they're a non-responder, we carry that through for 450 days. So we automatically assume that they failed across that line. When we have patients, that are at that marker, some are complete responders, some are non-responders. So I don't think it's dramatically changed across the board. I think there's clarification for the data set as you go forward. I think what will be really exciting is as we get our hands on the data for past 450 days to see how those patients respond and duration of response. Another one?
This one here, yeah.
Carrie Smith, your release that identifies Rutherrin as an enhancer to immunotherapy just seems like a gigantic game changer, which will attract interest from every major pharma looking to combine and extend their patents. Is there any more color you or your doctors can add to this to help us to realize the whole potential? I think that the Theralase has had a flurry of press releases over the last 2 weeks. I think there's been about 6 releases or 5 releases, something like that. What I think we've shown is that Ruvidar is a remarkable molecule. So not only does it have effect in bladder cancer, so non-muscle invasive bladder cancer, CIS, and papillary Ta/T1, it has effect in brain cancer, glioblastoma multiforme. It has effect in non-small cell lung cancer. It has effect in blood-based cancers such as leukemia, lymphoma, multiple myeloma. It, based on the efflux of the drug, it does a number of things with immunotherapy, with chemotherapy, making the drugs more efficient on dealing with bacteria. It makes BCG more effective. So it's a remarkable molecule, and I think that the company has fantastic technology. Our main focus, though, is still bladder cancer. Like, we'd like to get that one across the line and finish it. For a lot of these applications, What it's just showing is the diversity and the utility of that molecule. I think that that molecule for some other activities, if the company's not properly capitalized in which to do it, which it may or may not be this year as we move forward, would be to partner that technology. So there's certainly opportunities to work with other like-minded organizations. Obviously, if the company can become better capitalized, it can keep significantly more value in the organization by developing it. But there's a lot of costs involved there. So now you're going to get into a cost reward and cost risk analysis. Some of the technology will develop, some of it we're going to partner.
Perfect. That's great. We have a number of questions along these lines around uplisting and reverse split. So so a number along this line of reverse split and uplisting.
Anonymous attendee, when could we expect the reverse split and NASDAQ listing? Well, I'm opposed to the reverse split just simply because I don't think it works. I think that investors generally don't look at market cap, they look at stock price. And if the stock, and I've seen it multiple times where a stock does a 10-to-1 reverse, and I can give you numerous examples. There's a lot of pot companies that have done this, where the stock is a 10 cents, and then they do a 10-for-1 split, and they take it to $1, and it just starts sliding down again. So it comes back down to the 10-cent range, and all that happens is you hurt the shareholders. So I don't think it's wise to take a TSXV company at this point and go headlong into the ocean. For NASDAQ, if you're not prepared for it and you're not properly capitalized. So I'm not sold on a reverse split. I think that I have seen one example where a reverse split worked, but that was on a drug that was already approved, and they turned around and they did a different mode of administration to the body for the same indication. So they had a very clear commercial opportunity for it. I can give you 20 examples where it didn't work. So I don't think it's fair to shareholders to cut down their amount of shares when the company is so close to realizing its value.
Perfect. This is a question here. I think we've answered this in one of our recent press releases. This one, we actually have answered it. Do we have any results?
Trader One, do you have any results from patients post 450 days as to whether they are still cancer free? I can't answer that.
Let me slide down here. So we just have a number of questions. So we're just choosing. Some of them are, many of them are similar. So I just want to find out which ones. Yeah. So another one about the. Let me just this app. This is. Oh, go ahead. Yeah, please. Yeah. Mm-hmm.
Why is there a decrease in complete response over time?
It's if you're looking at the subset, this is a very serious disease with no good outcomes. Okay, bladder cancer, you're dealing with carcinoma in situ, which is a red shag carpet which spreads across the inside of the bladder. Quoting a good friend of ours, Dr. Michael Jewett, the red carpet is very hard to resect, so it's very hard to remove. It's high-grade disease, if you saw from Matt's presentation, has a very, very high recurrence, very, very high progression rate associated with it. And that's just the nature of the disease, right? With the carcinoma in situ, non-muscle invasive bladder cancer, BCG unresponsive population, This is a very, very difficult population. So what the FDA has done is they said, we don't have any good answers for this disease, so we're going to challenge industry to come up with solutions for it. So what happens is that the disease has a high recurrence. So it could be that the disease is not completely destroyed because it's lurking in deeper layers below the urothelial layer. It could be hidden in other areas of the body, either upper tract or lower tract. It could be in the kidney, the renal pelvis, the ureter. There's quite a bit of urothelial cell carcinoma in the ureter. It could be in, for men, prostatic urethra. It could be in the urethra. It could be in the bladder neck. So it can hide a lot of locations. As I spoke earlier, as long as the drug is absorbed and light activated, it will destroy that cell. Okay, that's what we've been able to prove both preclinically and clinically. I think it's just the nature of the disease. With this disease, it recurs and it has a very, very high recurrence. Why does BCG work initially and fail 50% of the time after 1 year? Because the cancer is being reseeded. It has destroyed some of the superficial cancer, but not some of the deep lying cancer. There could be a lot of reasons for it. But if you look at every competitor that's out there, their data always trails off. Okay. So the biggest thing I think the CEO of Immunity Bio stated, initial response is good, but the true determinant of a effective cancer drug is duration of response. That's what we've been able to demonstrate. So as far as our duration of response, it's world class. That answer your question?
Yeah, I guess part, it's, in other words, it's the ones that are complete responses is referring to the cells that the chemistry is able to get to, not the cells that.
Well, CIS is a superficial cancer, so it is with the urothelial layer. It's not into the deeper layers of the bladder, and it's certainly not into the detrusor muscle. So it's certainly not muscle invasive at this point. You're right, the drug must be absorbed, at the different levels, which is why some of the work we've done, the optimization in August 2020, which showed that we increased the volume of the drug going into the bladder, which is the study drug volume, the study device volume, because we wanted to increase the ability of the drug to interact with the urothelial cell carcinomas. Any other questions?
One right here from Christopher Horner again.
Your voice. Yeah. Christopher Horner, why do you think there is such a market value discrepancy, your slide 20, between TLT and its competitors, some of which are showing less efficacy than TLT's treatment? I think that I don't know, but if I would have to hazard a guess, I would say that those organizations trade on a much larger market. They trade on the US market. Those are all US listed companies. So they're NASDAQ listed. They're in the world's largest market, and they have 10 times the number of investors. I also think that the US market is very specialized because there's a lot of funds that do nothing but healthcare and life sciences. In Canada, we're pretty much special situations. I think that there's no funds very few funds that focus only on life sciences and healthcare. So I agree that the opportunity is to get to NASDAQ in the US market. The idea is, how do you get there? Okay. I think that the company has the ability and it has the technology to get there organically. And we need to get to a $2 US stock to get onto NASDAQ organically, not do a reverse split, not do crazy machinations with shareholders' investment, but to speak to like-minded organizations who are willing to help develop the technology or which are able to help fund the technology, and we get to NASDAQ organically.
Just a couple more questions we're going to take. Oh, yep, go ahead.
Sorry.
There's a number of patients who didn't respond at the very beginning. And it seems like a very high. Is there any research done to identify why that's happening?
Well, I think that if you look at all the competitors out there, they also have a high percentage of patients who don't respond. Okay. So again, if the drug doesn't get to the cancer cells of interest or it's not activated, there are cases where the disease can be on what's called the ureter orifice. So it can be right on the connection between the ureter and the bladder. So it may be partially in the ureter, partially in the bladder. Theralase may be able to destroy the disease on the bladder, but is not going to be able to destroy the disease in the ureter. So it can be reseeded. There are some patients that have gone through multiple treatments. We have, for example, in our data, I think approximately 50% of the patients we've treated have failed other therapies, chemotherapies, immunotherapies, oncolytic viruses, and they've gone on to be successful under our study. Okay, some do, some don't. So I think it's an understanding of the disease state that why do some patients respond and others don't? I think that it would take more research, more understanding, more in-depth analysis, but we can be pretty assured, based on the work that our Arcadi's team has done, that if the drug is absorbed and activated either through light or radiation, it will destroy the target.
Perfect.
So something blocking.
It could be deeper. Yeah.
Because you can't use really the examples of the ureter that can't be applied to like the majority of.
It's an example, yes.
Uh,
So with the bladder, like you understand with your bladder, that you've got your kidneys, which are filtering your blood. They're bringing urine down the ureters, they're dumping it in the bladder, okay? Which we void the body through the urethra, okay? The bladder collapses in on itself. So it has undulations or folds, okay? The bladder is expanded with the study drug. And so to absorb the drug, the patient may not be able to hold the study drug volume. They may void uncontrollably or they don't want to hold it. And the drug may not be in contact with the urothelial cell carcinoma long enough. So if there's still disease remnants there, then it's not going to be activated. Same thing with the study device volume. If there's diverculi, if there are areas where the light is being shielded, such as the bladder neck, where the cystoscope is sitting, a metal tube, it can block that. So there may be a couple of things. I think it would have to take an analysis of those to see why we're not as effective as some of the other patients.
Because if you could solve that problem, that would greatly double the response.
It's noted.
Great. I think just one more, and just to let everybody know, thank you for the questions, and thank you for the questions as well. There's still some good questions out there. I'll take a look at them and I'll review them and I'll respond over the next few days to those questions. But thank you. So last question.
Sure. Michael Bashira, what is the plan with Theralase? Does management see the company becoming a producing pharma company or sell the company in the future? I think that everything is on the table at this point. The company, and we've made no qualms about this, needs to be properly capitalized in order to execute on its technology to deliver shareholder value. That can come in many forms. It can come in the form of financing. It can come in the form of royalty deals. It can come in the form of partnerships or distributions. So I think everything is available for the company at this point. But again, where the company is going at the moment is to complete its non-brokered private placement, become base shelf eligible. And then as it moves forward to completion of its phase 2 clinical study, which are our main asset, that's where you could start looking at different organizations to partner it. Because Theralase is a great technology company. It has world-class technology. It has world-class data. We are not a distribution company. We wouldn't have the distribution of a Merck or a Pfizer or AstraZeneca or whomever you want to pick. So if you want to get your drug around the world to 75 or to 100 countries, you're better off to partner it, because for us to build that from scratch would take centuries and we'd have to buy and hire a lot of people. Why not partner it with who's good at it? So Theralase is a technology company. We have world-class technology. We plan to take it to a certain level in order to build shareholder value. But at some stage, if we wanted to go to every corner of the world, it would have to be partnered.
That's great.
Okay.
That's great. Thank you very much for joining us. And thank you again virtually for joining us as well. We're going to close down the meeting. And again, we have some refreshments outside. Have a good afternoon. Thank you.
Thank you. Hold on.